Hi, I'm Nasreen Bushi. Today I'm going to give you a demo on the topic photosynthesis in plant. Before I start this topic, I want to share something related to my qualifications. I have completed my master degree in plant pathology from Aligarh Muslim University with the same subject institute. So I recently submitted my PhD degree, and besides this, I qualified ICNET exam of plant pathology. I'm the awardee of two gold medals for securing highest mark at both department and university levels. And currently, I'm working as assistant professor of plant pathology at local university Saharanpur. So let's start this topic. Why the plants do photosynthesis? Because we know plants are the autotrophs and they need to synthesize their own food by utilizing sunlight energies, and the food is glucose molecules. So we can compare the plants from ourselves, like we human beings and animals. Basically dependent and directly consume plant-based products or animal-based products which is already rich in glucose molecules. So here we can understand the definition of photosynthesis. The photosynthesis is usually completed by utilizing solar energy, and how this solar energy is trapped due to the presence of this chloroplast organelle in the leaf, which have a characteristic important characteristics, presence of light harvesting pigment like chlorophyll A, B and carotenoids. So this one glucose molecule is synthesized in the presence of sunlight by consuming 6 carbon dioxide molecule from the atmosphere into the stomata. 12 water molecules enter from soil into the plant root. So this is how this reaction is completed. So one glucose molecule after produced as a product, and 6 water molecules evolve out during transpirations, and 6 carbon dioxide molecule goes out into the atmosphere from the same part of the stomata. Now the photosynthesis, here we understand photosynthesis is completed in two ways. The one way is called light dependent process, light reaction or heat reaction, which usually completed in the granular region of the chloroplast. And the second part is called as light independent phase, and you can also call as dark reactions or Calvin cycle, usually completed into the chloroplast of the into the granular stroma region of the chloroplast. So here this picture clears you the mechanism or the two phases like light reaction, dark reaction is completed with the help of this chloroplast organelle. So here this is the chloroplast organelle. So this chloroplast organelle has two parts. One, this blank portion is called as stroma region. In this blank portion, there is a stacking of disc-like structure, which is called, one disc-like structure is called thylakoid, and whole stacking is called as grana. So grana, this is called grana, and they are green in color because they are highly rich in pigments, light harvesting pigment, as I already cleared you. This one is called as thylakoid. So here we have to focus for light reaction on this disc-like structure. This disc-like structure called thylakoid has two parts. One outer part is called as thylakoid membrane, and the part which is present below this membrane is called thylakoid lumen or space. So here this thylakoid membrane is you can see here this thylakoid membrane is embedded with lots of protein like photosystem second, various electron carrier molecule, plastocyanin and plastocyanin and photosystem first. And one important part is called ATP synthetase. So this ATP synthetase is basically utilized to produce ATP rich energy molecule, and by completing in two steps. So first what happened in this light reaction, the photosystem second is initiated or trap the light energy of wavelength 680 nanometer to extrude out the electron. This extruded electron is passing various electron carrying molecules and to initiate the electron transport chains. At the same time, photolysis of water is also take place. So this photolysis of waters release oxygen atom, hydrogen ion, and two electron particles. So these two electron particles, what happened with this? These two electron particles have to compensate the electron which is lost during the capturing during the this photosystem second event. And one more thing happened here. This they when the electron passing from one electron carrier molecule to another electron carrier molecule during electron transport chain release lots of energy. So this release lots of energy is sufficient and utilized to pump the proton ion which is present in the stroma region, this stroma region, from this stroma region into the thylakoid lumen or space, in this thylakoid lumen and space. So this is the first way how is the proton ion accumulated into the thylakoid lumen. And the second way, the photolysis of water which release the proton ion, this proton ion is also accumulated into the thylakoid membrane. So this is how the proton ion accumulated into the thylakoid lumen by two way. One is the photolysis of water, and second way during the passing of electron from one carrier molecule to another carrier molecules. So this is how in thylakoid lumen proton gradient is developed. This developed proton gradients activates the ATP synthetase. This ATP synthetase is also embedded into the thylakoid membrane, which facilitate the diffusion of proton ion from high concentration site towards the lower concentration site. That is the region from thylakoid lumen to the stroma region. So this break of proton gradients liberates a lots of energy which utilized to combine inorganic phosphate with the adenosine diphosphate to produce ATP. So this is how the ATP molecule, that is adenosine triphosphate molecule, is generated by the breaking of proton gradient which is developed into the thylakoid lumen.